Good afternoon, everybody, and welcome to the second of our spring semester scientific colloquia. It is a great pleasure to be introducing today's speaker, Rick Henson. Before I introduce Rick, just a couple of housekeeping issues. As previously, could I encourage you to use the chat to ask questions of Rick. Don't wait until the end to send in your questions as they come to mind. Please just communicate them on the chat, and I will go through them and moderate the questions at the end, and I will ask Rick each of the questions to the best of my ability, and he will answer to the best of his ability. And so that's what we will be doing. An innovation compared to previous events, though, is that at the end of the formal proceedings, that is, which will come to an end at around about 1 o'clock our time, there will be an opportunity to switch links. The link to switch to will be in the chat box, and if you click on the new link, you will emerge into a chat room, a regular Zoom-like chat room, with the opportunity to talk to Rick more informally, and for him to actually see who it is who he's interacting with. And we'll try and keep that kind of backstage arrangement for about 20 minutes or so. Many thanks to Rick for agreeing to do this. Fingers crossed that it works. So without further ado, let me introduce Dr. Rick Henson. Rick obtained his first degree and his PhD from the University of Cambridge, graduated with his PhD in 1996. He then went on to postdocs and a variety of increasingly senior research fellow positions at UC London, University College London, and was based for much of that time in the Functional Imaging Laboratory in the Institute of Cognitive Neuroscience. And in the interest of full disclosure, he and I collaborated pretty closely over much of that period. Since 2004, he's been at the Medical Research Council's Cognitive and Brain Sciences Unit at Cambridge. He's currently the acting director. Oh, I'm sorry. You stopped being acting director, didn't you? You're now deputy director. Is that correct, Rick? Sorry.
Yes.
Also is Professor of Cognitive Neuroscience at the Department of Psychiatry at Cambridge. And Rick, I think if there's anything that could be considered to be a polymath in cognitive neuroscience, then that is Rick. He's extremely well known for his work, both empirically and theoretically, in the fields of episodic memory, attention and perception, and working memory. He's also extremely well known for his seminal methodological contributions to functional imaging, and also for his work along with colleagues in Cambridge, putting all of these developments to good use in the study of cognitive aging. And today I'm very pleased to introduce him, and the topic of his talk is going to be relating age, brain and cognition, results from the Cambridge Centre for Aging and Neuroscience. So without further ado, over to you, Rick.
Thanks, Mick. Yes, that is my title, so I can move on. So I don't need to tell this audience that the population demographic is shifting towards older ages, and therefore maintaining cognition in old age would help people function independently for longer. But of course, this requires an understanding of how to keep the brain healthy in old age. And there have been many cognitive neuroscience studies of aging over the last 2 or 3 decades, but they've tended to use rather small and sometimes unrepresentative samples. For example, older people responding to adverts to take part in experiments who are often biased towards being more physically and cognitively able than the average older person. And these are the reasons why in 2010, 10 years ago now, over 10 years ago, we tried to recruit a more population representative sample of adults across the whole lifespan from 18 to 88 years of age, who we could obtain cognitive brain data, that's MRI and MEG, genetic and lifestyle data. and hence CamCAN was born. If you want to read more about CamCAN, this is the website. So, for example, PDFs of all the papers or the studies I'll talk about today can be obtained from there if you want to follow up any details. In brief, we went round and knocked on the doors of nearly 3,000 people, and they had to opt out to say no, they didn't want to take part, which makes it slightly more representative than the typical study. And if they agreed, they had a 2-hour home interview where we took basic lifestyle and questionnaire data. And then we subsampled 100 per decade from 18 to 88 to come into the CBU for, in total, about 7 hours of cognitive testing over 3 study visits. And during which time they also had an hour of MRI with a large number of different sequences, as well as half an hour of MEG. A further 40 per decade, the CC280, came back for a 4th and 5th visit to do some more specialised task-based fMRI and MEG. But I'll be talking mainly about the CC700 in this talk. And those data from the CC700 we made available to the world. So if you want to access the data from CamCAN, you can go to this site here. And as long as you consent to a few requirements about data sharing, then we will give you the data. And so far, I think nearly 900 researchers around the world have downloaded these data and hopefully found them useful. But today I want to basically give you an overview of some key findings from CamCAN, sort of smorgasbord of tasty tidbits, and I'll only describe each study very briefly. Happy to expand on the study afterwards, or you can look at the papers. And the first 3 studies will all to do with functional connectivity. The first one will look at the issue of whether functional connectivity, as measured with fMRI, is a state or a trait of an individual. I'll then talk about our realization that it's very important to control for vascular effects when you use fMRI to measure functional connectivity, because vascular health, as you probably know, decreases as we get older. And then I'll talk about a very recent study where we used MEG instead to look at dynamic functional connectivity of periods over 100 milliseconds or less, and looked at how that affects changes with age and how it relates to cognition. I'll very briefly then talk about the first genetic study from CamCAN about the ApoE polymorphism. And then I'll finish by talking about 3 studies that address cognitive theories about aging, such as dedifferentiation, compensation, and cognitive reserve. With functional connectivity, this was a study published a while ago now by Linda Geerliks, who was a postdoc on CamCAN but now runs her own group at the Donders in the Netherlands. And you're probably aware that resting state fMRI connectivity is often assumed to be a trait of individuals, such that you can distinguish, for example, patients or controls based on their functional connectivity. And it is true that when you average across individuals, the same core networks tend to occur invariant to the task or invariant to the state. However, just because the same networks occur when you average over tasks does not mean that individual differences, for example, due to age, are invariant to state. For example, a particular functional network might be expressed more in young than old individuals during rest, but expressed less in younger people than old individuals during a task. And that's what we try to address in this study. So in CamCAN 700, there are actually 3 states of where we can measure measure functional connectivity. There's rest, and there was a sensory motor task, which I won't talk about today, and also a movie task where people watched a Hitchcock movie for about 9 minutes. I'm not going to go through the details of the fMRI pre-processing, although I can if you're interested in questions. But we ended up with looking at connectivity within and between 16 large-scale networks. And we looked at the factorial effects of state, whether you're at rest or watching a movie, a linear effect of age, and most importantly, the interaction. So does the effect of age depend on the state you measure it in? And these are the 16 networks. And here is the main effect of state. So this is a connectivity matrix where the 16 large networks are on the axes. And I'm going to roughly divide these into sensory motor networks, the first 3 here, subcortical networks, the next 4, and the remainder just coarsely going to talk about them as being higher order networks. So in here, a warm color means that the functional connectivity on the diagonal within or on the off diagonal between networks, a warm color means greater connectivity when you're watching a movie than at rest. And conversely, cool colors mean greater connectivity at rest. And a little dot means that the connectivity survives Bonferroni correction for multiple comparisons. So these are quite reliable effects. The first thing to note is that when you're watching a movie, most connections tend to increase. So the warmer colors here, at least subcortical and higher order networks. As an aside, I was quite surprised to find that the connectivity within and between the sensory motor networks actually decreases when you're watching a movie. Because I would have thought they might increase because of the fact that a movie tends to have correlated visual and auditory input. So you might expect the sensory cortices to become more correlated in their activity, but actually they become less correlated. And this could be because they start to segregate into processing their own modality-specific information, and maybe that reduces connectivity compared to at rest, where they might be dominated by large endogenous rhythms across the whole cortex. Anyway, that was an aside. The more interesting effect is the effect of age. So we actually tested the interaction, but to make that clearer, I'm showing the simple effects of age for the 2 states, rest on the left and movie on the right. And again, just to look at global patterns, we tend to find increased functional connectivity between networks, certainly subcortical networks, as you get older, and that replicates what other people have reported as well. But importantly, this also was modulated by the state. So when people were watching a movie, actually there was decreased connectivity as people got older. That's the blue colors here in the movie, but that was less pronounced at rest. And conversely, in the sensory motor networks, there was a negative effect of age at rest, which wasn't apparent during the movie watching. In other words, there were quite a few connections in which the effect of age depended on what state the connectivity was being measured in. So the overall pattern, as has been found in many groups, including in Dallas, is that aging tends to increase between network connectivity and therefore decreases functional segregation. And that's something I'll come back to later in the talk. But for this study, the important point is that these age-related effects depend on the state you measure them in, such that functional connectivity is not necessarily a stable trait of an individual, and no particular mental state, such as resting, is uniquely suited to study individual differences in functional connectivity, at least we would claim from these data. We did actually look at trying to get correlations between these functional connectivity measures and our multiple cognitive measures. These are the early years of CamCAN. And in general, we only found weak or non-existent correlations between functional connectivity measured this way and cognition. And again, this is something I'll come back to when I've dealt with the issue of vascular health. So in a later paper, we looked at vascular health in CamCAN, and we had a large number of measures like blood pressure, ECG from the MEG recording, which we can measure heart rate variability, etc. And if you take the first principal component of these vascular measures, we actually did this measurements on 2 occasions. And if you get this first principal component of vascular health, we find it's very reliable. So across 2 sessions, there's very high correlation, suggesting it's a robust measure or reliable measure. And if you look at this vascular health against age, it tends to decrease. And that's not surprising. Vascular health tends to get worse as you get older. What's important is that we found a strong positive relationship between functional connectivity measured by typical fMRI analysis and vascular health, even after removing the effect of age. So having adjusted for the effect of age, there's still a positive relationship between vascular health and connectivity. So if vascular health decreases with age and vascular health contributes to functional connectivity, some of the effects we're seeing in our fMRI may be due to vascular effects rather than true neural connectivity. So what can we do in this case? Well, in that paper, we tried various methods to remove this correlation with vascular health. And the only thing that we found the most effective was just to remove the mean functional connectivity over all connections. This means that you can't look at the overall effect of age on connectivity in general, but you could still talk about whether age affects some connections more than others. But if you don't want to adjust the data, another option is to keep the data unchanged, but have a more sophisticated model of functional connectivity, which can take into account age-related changes in vasculature. And for that, we use something called dynamic causal modeling. And this is now the work of Carmen Stefanoff, who is another postdoc on CamCAN, who now runs his own group in Cambridge. So in brief, resting state DCM, or dynamic causal modeling, I can hopefully express quite simply as having three parts. The first part is the activity model, and this assumes that when you're resting, your power spectrum of your activity, or your neural activity, can be expressed simply by an exponential function with two parameters, alpha and beta, the amplitude and the exponent of this exponentially decreasing function of frequency. So a very simple power spectrum model of neural activity at rest. The second part is the connectivity model, which you have to specify, and we chose three large-scale networks shown on the screen, and we kept them together, and that was our model where we're going to try and infer the connections between these and how they change with age. And the third and final part of DCM is the hemodynamic model, which is the most important part for vasculature. And this is a biophysical model based on differential equations, using some parameters that are based on what we know from measuring, for example, in animals, the relationship between neural firing, vasodilatory signals, change in blood oxygenation volume, flow, etc. And so this has many parameters, although you can project them into a three-dimensional space. So basically, most of the variability in the hemodynamic response can be expressed by three parameters that govern the relationship between neural activity and the ultimate signal we measure with fMRI. So these are the three parts of DCM, and then we can ask, does age affect the neural, the connectivity, or the vascular parameters? And here are the results. So I mentioned there are three vascular parameters, and on the y-axis here is the effect of age, and transit, decay, and epsilon are these hemodynamic parameters. The three different bars are the three different networks, but you can forget about that for the moment. The most important thing is that there was an effect of age, at least in two of the networks, on the decay parameter of the hemodynamic model. So that shows that age affects the neurovascular coupling. We didn't find any effect of age on the two neural parameters, reflecting the amplitude and exponent of this exponential neural decay. And so no effect of age on those. But we did find effects of age on the connectivity parameters, at least in some of the networks. So basically, by separating out the hemodynamic and the neural parameters, we can now estimate connectivity at the level of neural connectivity rather than direct fMRI connectivity. And so we found that age affects various parameters, and then we can then ask, does this neural or affective connectivity relate to our cognitive measures in CamCAN? And for that, we use canonical correlation analysis, or CCA, but basically that's just a way of seeing whether some linear combination of, in this case, cognitivity parameters relates to some linear combination of our cognitive scores. And indeed, we found a highly significant first, what's called canonical correlation or canonical variate, where some combination of connectivity parameters related to some combination of cognitive parameters. Most importantly, if we plot an individual's score, their cognitive profile, against the connectivity profile, we found a significant relationship that actually increased as you got older. So in other words, the older you were, the more your cognitive scores depended on your neural connectivity profile as measured by DCM. And when we repeated this analysis using a more traditional measure of functional connectivity, namely just the Pearson correlation coefficient between the raw fMRI time series, there was a much weaker canonical variate and no evidence that this relationship between cognitive profile and connectivity profile was moderated by age. And so this suggested that by using DCM to uncover the neural connectivity allowed us to be more sensitive to cognitive effects of aging than a standard Pearson correlation coefficient. To summarize, standard resting state functional connectivity as measured by fMRI is affected by vascular health. And once you allow for vascular contributions, the relationship between that measures of connectivity and cognition gets stronger. So if you want to study the effects of age on neural connectivity, either adjust your data, for example, by the mean functional connectivity or some independent measure of vascular health, or have a more sophisticated model like DCM, where you can look at the effects of age on hemodynamic and neural connectivity parameters separately, or simultaneously, you can estimate them. Or the third thing I want to turn to now is to use an imaging technique that bypasses such neurovascular coupling, such as MEG, which is a more direct measure of neural activity. And the other thing we can do with MEG is actually look at changes in connectivity over time, because everything I've talked about so far has been static connectivity across the whole 9 minutes of rest or movie watching or whatever. This brings me to the last study of this section, using MEG. So we actually started with fMRI, and we took various methods that have been proposed in the literature to look at dynamic functional connectivity using fMRI. In other words, to look at how connectivity might change over windows of 30 seconds or 60 seconds throughout a recording. But when we did some simulations, this is work by Brio Lehman, we found that actually once you take into account effects of age, for example, on the hemodynamic response or on the autocorrelation of the time series, then these methods for measuring dynamic functional connectivity with fMRI, they just failed. So they failed to recover individual differences, for example, due to age, when those differences could occur on other components of the fMRI signal. And that's why we didn't think it would be very fruitful to look at dynamic functional connectivity using fMRI. So when we turn to MEG, obviously we suffer in terms of spatial resolution, but what we gain is temporal resolution. And we can look at these connectivity states that wax and wane of the order of 100 milliseconds during a 9-minute rest period. And for that, we used Helen-Markov models, which I'll describe very briefly in a moment. But the nice thing about this is we can also bypass these age-related effects on the vascular response. And this study is now in press, Neurobiology of Aging. It's led by a postdoc in my group called Ronnie Tebon. And so we took the 595 resting state MEG datasets in CamCAN, who also had valid MRIs and cognitive data, etc. And we looked at 8 network states. We used 8 because that's a number of states that's been used in previous studies from the Oxford group, who've developed this method of hidden Markov models for investigating resting state connectivity. And then having estimated these 8 networks, we related them to age and cognition using the same CCA approach that I told you about for fMRI in the study of Setvarnov et al. So here are these 8 what are called microstates or HMM states. So the brain shows you the rough distribution associated with that state over the brain. And then the bars reflect some dynamic properties of those states. Things like FO, the fractional occupancy of that state throughout the 9-minute period, or the mean lifetime of a particular state. So obviously these dynamical measures of how long someone spends in a certain state or how often they switch between states are all interrelated. If you spend more time in one state than another, that's necessarily the case. If you spend more time in one state, you have to spend less time in another. So these variables are all interrelated, which is partly why we use CCA I'll describe in a moment. But shown here in the bars is the effect of age on these dynamic measures for each of the states. And the broad pattern is that as people got older, they spent longer in these frontal temporal parietal states and less long in these early visual states, even though this is now at rest with eyes closed. We also then did CCA, so we related this set of 32 dynamical measures of states to our multiple cognitive scores. And we found, again, a strong canonical correlation for the first canonical variate. And these are the loadings across the cognitive scores. And this resembles impaired fluid intelligence. So these 2 bars that are positive because these are the 2 tests that measure reaction time. All the other bars are negative, measure accuracy. So this general pattern of slower and less accurate is associated with reduced fluid intelligence as a sort of common component to all these tasks. And it didn't load so highly on the tasks that we think are more to do with crystallized intelligence. So we interpret this as a fluid intelligence measure that was highly related to these brain measures. And again, looking at the effect of age, we again found that the older people get, the more their cognitive ability or their fluid intelligence was related to their the dynamical properties of their different brain states as measured with MEG. So the lighter colours here are older people. And I don't know how clearly you can see this, but the slopes are getting steeper in older people. Obviously, this was done in a continuous manner, and I'm just showing you discrete ages to make it easier to understand the interaction effect. To summarize, age increased the occurrence of these higher order networks, how often they occurred, and decreased the occurrence of lower order networks. And this was associated with worse fluid intelligence. And this relationship was moderated by age, being stronger in older people, like we found with fMRI. Now, the fact that the dynamic functional cognitivity pattern that was associated with the fact this pattern of increased frontal temporal parietal involvement compared to early visual involvement, the fact that's associated with worse fluid intelligence in very hand-wavy terms doesn't seem to be consistent with the idea that this reflects compensation in some way. I'd argue it's more likely to reflect reduced neural efficiency. And I'll come back to this idea of compensation versus efficiency later in the talk. So that's all I wanted to say about functional cognitivity. Let me briefly describe a genetic study from CamCAN. And this came out last year, and it's a registered report. So we specified what we do before we deblinded the phenotypic data and the genetic data from CamCAN. And we're focusing on the ApoE polymorphism. As you're probably aware, the epsilon 4 or E4 allele that's part of the ApoE gene is associated with cognitive decline in old age, and particular increased risk for Alzheimer's disease compared to the more common E3 polymorphism. And there's a hypothesis called the antagonistic Plyotrophy hypothesis that claims that although epsilon 4 might increase your risk of dementia in old age, it might confer benefits when you're younger. And for example, people have found that younger people with epsilon 4 show increased activity as measured with fMRI during cognitive tasks. And so maybe it helps you when you're young and impairs when you're older. And that could contribute to the prevalence of this epsilon 4 gene in the population. Well, that's not the only reason, but maybe that's one reason why this still remains a relatively common variant. So this hypothesis predicts if the blue line is the more common epsilon 3 group, so this is some measure of cognitive or brain health against age, the antagonistic Plyotrophy hypothesis says that, yes, we know that if you have epsilon 4, you suffer in old age, but maybe you have some benefit earlier in life. There's also another polymorphism, the epsilon 2 variant, and that has been claimed to be neuroprotective in old age. And that's the green dotted line here. So it may be that when you're older and you have this variant, maybe you're more resistant to decline of the brain or of cognition. So we wanted to test this idea by looking for an interaction between age and ApoE status using a polynomial expansion. And we looked at this interaction on 6 outcomes, which are all based on prior studies. So we looked at the effects of this interaction on fluid intelligence, episodic memory, hippocampal volume, white matter integrity, default mode functional cognitivity using fMRI, and generic functional cognitivity from MEG, because each one of these has a prior published study that's claimed that ApoE affects that outcome. Now, you might be thinking that this is a very small sample for genetic analysis, only about 600 people. But remember that ApoE has a very high penetrance. And actually, if you took the effect sizes from the previous literature, so I'm putting here power against effect size. The vertical black lines are the effect sizes numbered according to the legend on the left from previous studies. And you can see for most of them, if these effect sizes were true, even with 600 people, we'd have close to 100% power. The green curve is for detecting effects of epsilon 2, which is even rarer. The red curve is for epsilon 4, and the blue dotted line is a dose curve if you assume a linear relationship from E2 to E3 to E4, then we should have a reasonable power for most of these outcomes, if these effect sizes are true. Unfortunately, we didn't find any evidence for an interaction between age and ApoE status in any of our 6 outcomes. So each colour here is a different genetic group, and they have the effect of age on these 6 outcome variables, and the strong effects of age, but no evidence for interaction with genetic status. Indeed, if we actually use Bayes factors, we find compelling evidence for the null hypothesis that there is no interaction. In other words, we find evidence against the antagonistic plyotropy hypothesis. So that's just an example of the kind of studies that we're now doing. We're also part of a larger life-brain consortium of cohorts in Europe, where we're going into the tens of thousands of people with MRI and cognitive scores, where we can look at sort of more polygenetic risk scores. Anyway, turning to the last part to talk about cognitive theories. So this is a study led by Suzanne Demouge and my long-term colleague Roger Kiewit, who's now back at the Dondas as well, leading his own group. And as I'm sure you're aware, the idea of differentiation comes from developmental psychology, and it's the idea that as kids get older, there's a decreased correlation between their performance on different cognitive tests. If you like, their cognition becomes more specialized as they get older. And the opposite has been hypothesized to happen in old age, called de-differentiation, the idea that as we grow older, all our cognitive abilities start to become more similar, and in other words, show increased correlation between tests. Although other people have argued that's not always found. You can look at the same thing with the brain. You can take the dominant white matter tracks and look at their integrity using diffusion-weighted imaging, and ask whether the correlation between these increases or decreases as we grow older. And it's been claimed by a study by Cox et al. That the correlations increase as we get older, so de-differentiation of the brain's white matter. And that's similar to what I showed you with functional cognitivity, that the networks become more correlated with each other, more similar. But what people haven't looked at prior to this study was when there's any differentiation of the relationship between brain and cognition. So in other words, does the covariance between brain and cognition increase or decrease as we get older? For example, if there were some kind of functional compensation that some people make, or maybe people differ in their amount of cognitive reserve, then you might expect the relationship between brain and age to become weaker, to differentiate. Conversely, if you believe that the brain maintenance hypothesis, that your cognitive health is directly related to your brain health, then you might expect no change in this covariance across the age, across adult age lifespan. So we took, again, data from cognitive measures. We also in this paper looked at grey matter, but in the interest of time, I'm going to focus on the white matter results across 10 large white matter tracks from an atlas. And we use something called multi-group structural equation modeling to test this idea of differentiation. So basically, we split the CamCAN sample into 3 age groups, young, middle age, and old. And you can fit a structural equation model to each group separately. And then you can, where the parameters can be free, and you compare how well that fits compared to a model where the parameters constrained to be equal across the 3 age groups. Or at least the covariance parameters between brain and cognition can be constrained to be equal across groups. And you ask whether that model is significantly worse than the model with the parameters being free or allowed to vary with age, despite the additional degrees of freedom. And then having done that in a categorical sense, you can look at the effect of age using a moving window approach to structural equation modeling. So that might sound a bit obtuse, so let me try and explain. But starting with the cognitive factors, we did confirmatory factor analysis to divide these into 3 main factors: language, fluid intelligence, and memory. And this is the resulting structural equation model. And if we look at the latent variables of language, fluid intelligence, and memory, you can see that language is relatively stable across the lifespan, memory decreases, and fluid intelligence decreases quite precipitously in old age. But what we're interested in is these 3 parameters here. These reflect the covariance between these latent variables estimated from the observed data. And the question is, do those covariances change with age? And when we fit a model where they're constrained to be equal, versus those where models were allowed to differ, we found the model that they're constrained to be equal was preferred. In other words, it's more parsimonious. You didn't gain anything or sufficient fit by adding more free parameters. So there's no evidence that cognition changes, at least in this data, as a function of age. No differentiation or de-differentiation. How about the white matter? Well, here are the 10 white matter tracks I mentioned. And so we actually used exploratory factor analysis to find that a 3-factor model was again favored over a 2 or a 4-factor model, or a 1-factor model. These latent variables tended to all decrease as we get older, but the key question was, do the covariance between them change with age? In this case, we found a different result to cognition. We found that the model where they were free was a significantly better fit. And this suggests that the covariance between the white matter does change with age. But this is the most important part. So this is now looking at the 9 parameters, the 9 covariance parameters that relate these 3 white matter brain measures to these 3 cognitive measures. So we're now asking, in a model where we relate brain and cognition, does the covariance between the 3 cognitive latent variables and 3 brain latent variables, is that fixed with age or does it change with age? And we found that a model where they were free was a significantly better model. So now I explained more of the data despite the 9 extra degrees of freedom, or even 18 actually, across the 3 age groups. And if we look at use a moving window to look at what's changing, what we find in particular is that the relationship between this 3rd white matter factor, sorry, the 3 columns here are the 3 white matter factors, the 3 rows are the 3 cognitive latent variables. And what we find is the covariance between memory and this 3rd white matter factor tends to decrease as we get older. That seems to be driving this effect. And interestingly, the 3rd white matter factor included the hippocampal cingulum as a major tract. So, using multi-group structuration modeling on brain and white matter, we find no evidence for cognitive differentiation, contrary to some previous studies, but actually consistent with other studies, such as the Lothian birth cohort from Deary and colleagues. We did find differentiation in the white matter of the brain, at least the 3 factors we found, which again is different from Cox et al., but they only used a 1-factor model. So I think allowing a more flexible model of the white matter might be the reason for that difference. But the most novel part was evidence for differentiation between brain, or at least white matter, and cognition. And this seems to be driven by a decreased relationship as we get older between memory and this 3rd white matter factor, which, as I mentioned, loaded highly on the hippocampal cingulum. So what is the reason for this differentiation between brain and cognition in old age? Well, it could be because, well, firstly, people could be aging at different rates. So you get, as we know, longitudinal growth curves show quite different individual trajectories. Another reason could be that we're seeing a division into groups in old age. Some may have incipient dementia or pathology related to dementia, which hasn't caused them to be classified with dementia, because everyone in CamCAN passes standard mini-mental and ASAR tests of not having dementia. But maybe there's subgroups developing dementia, or maybe the subgroups who are able to compensate in some way or have increased cognitive reserve. And so I'm going to turn now to these issues of compensation and cognitive reserve. So this study on compensation was led by Alexa Morecambe, who now runs her group at the University of Sussex. And we were interested here on this hypothesis that when you look at task performance in a scanner, you often find the following result, where older people show increased activity in the frontal cortex. Here it's on the right in this PET study, when they're doing the same task. So is this increased activation in older people, is that compensatory? Does it help them recover some of their performance? And there is a general term for this pattern that you can see in quite a few studies of aging, where there's an increased activity in anterior brain regions, has been called PASA, the posterior to anterior shift in aging. And one thing that PASA may reflect is increased compensation in older people in frontal cortex, perhaps owing to loss of neural function or integrity in posterior parts of the brain. Is the prefrontal cortex compensating in some way? You can test this with many ways of relating behavior to performance activity, but here we wanted to test it using multivariate pattern analysis. And we want to ask, if this increased activation is compensatory, does it actually contain more information about the task, more information in terms of patterns of activity across voxels? And we used two very large ROIs, prefrontal cortex and posterior visual cortex, in two independent memory tasks in CamCAN. And I'll show you the results of the first memory task. But the key idea here is, if this prefrontal hyperactivation is compensatory, then it should carry more information about the task, or in this case, about whether an item will be remembered or forgotten. And we use something called multivariate Bayes to ask whether, if there is compensatory information in the prefrontal cortex, then when you add the prefrontal cortex to the posterior visual cortex, it should improve classification of the task. So there should be complementary or additional information in prefrontal cortex over what's in the posterior visual cortex, if compensation is correct. On the other hand, if the increased activation in prefrontal cortex is not carrying extra information, then it may be just an epiphenomenon, or maybe just reduced efficiency of the prefrontal cortex in older people. Maybe they need to just do more metabolic work to get the same neural outcome. So here are the two ROIs, and here is, on the y-axis, the mean subsequent memory effect, the mean activity in those ROIs related to whether you correctly remember an item or not. And what we found is in the prefrontal ROI, the red here, there was a significant increase in univariate activity as people got older. And that's just replicating the basic PASA finding. So in other words, on average, older people show greater activity associated with subsequent memory in this prefrontal ROI. But the question is, does that carry information? And we actually used three different measures of information. I'll just talk about one simple one in this talk, which is the standard deviation of the voxel weights. So the voxel weights are something you obtain when you do MVPA. It's how much each voxel contributes to a pattern. And generally, you might expect increased variation of those weights when there's more information. It's not the only measure or the best measure. We actually did more sophisticated measures in the paper, but this is the simplest one to explain. And what we find here is that this measure of multivariate information actually decreases as you get older. So this is contrary to compensation. Unfortunately, it means that this increased univariate activation does not carry more information about the task in this case. And instead, it may mean that this increased activation, this univariate effect, is actually just reduced efficiency of function in the prefrontal cortex. So to summarize, we replicated the PASA finding of increased univariate activity in the prefrontal cortex, but we didn't find any additional multivariate information. Indeed, we found less information in older people. And we think this is more consistent with less efficient function in prefrontal cortex when you get older rather than compensation. And finally, on a more positive note, I want to talk about our findings about cognitive reserve. This is work led by Dennis Chan, who is a neurologist in Cambridge and now has moved to UCL. So, as you're probably aware, cognitive reserve is really a catch-all term to explain an observation originally made by neurologists that some people can maintain their cognitive abilities in old age, even though a brain scan shows they have quite severe atrophy, and you might expect them to have, for example, dementia, but somehow they are performing well. So this gap between what you expect from their brain structure and their cognitive performance. One common factor associated with cognitive reserve is the level of education in youth. So people with greater education seem to be more robust to brain change as they get older. But here we were more interested in more modifiable factors, particularly midlife activities, because it's easier for a public health intervention, for example, to nudge people's behaviour in middle age than it is to completely reorganise the education system, arguably. So in CamCAN, we have a questionnaire called the Lifetime Experience Questionnaire, developed by Valenzuela and Sanchez. And we have this on all people, but I'm going to show you data now just from the 205 people in CamCAN who were above 65 years of age and had retired. And we defined their cognitive health by the Cattell test of fluid intelligence, although we get very similar results if we just take the first principal component across 12 more specialized cognitive tests. Here's the Leq questionnaire, and basically it's a questionnaire that divides your life into 3 time periods. You've got youth, midlife, and late life. And of course, everyone completed this, but just to make it clear, I'm only showing you results from the older participants who were retrospectively evaluating how much of these activities they did at the different stages of their life. Now, within each of the 3 life periods, there are 2 types of questions. There are specific questions that relate to that life period. So for youth, that would be education, and for midlife, that would be occupation. This questionnaire also has a set of non-specific questions that are repeated the same across all 3 life periods. So for example, how much did you see family and friends? How much physical activity did you do? How often did you play a musical instrument or whatever at each stage of your life, when you were young, middle-aged, or in this case, currently as you're old in this group. So these are the 6 scores I'm going to talk. Each of these comes from about 10 questions. So we have 6 large scores from a questionnaire. Now, if we correlate each of those separately with cognitive health, each of them has a positive correlation with cognitive health. But what we did was actually a simultaneous or multiple regression to see whether there are unique contributions of some of these life periods to cognition in old age, also adjusting for age and sex. And here are the results. So first, there was a negative relationship between cognitive health and your age between 65 and 88. So as you might expect, the older people did worse on fluid intelligence within this reduced life age bracket. We also replicated this positive relationship between your youth-specific activities, which is basically education, and your old age cognition. But what was interesting was there's also an additional unique contribution of midlife non-specific activities. So these activities outside the workplace, things like seeing family and friends, doing sport, etc. So they made a contribution beyond education, beyond age, and importantly, beyond the current activities people were doing in their late life, which didn't make a unique contribution. But that suggests that midlife activities may function like a kind of cognitive reserve, but I think to meet the precise definition of cognitive reserve, these midlife activities need to moderate the relationship between brain health and cognitive health. So on 195 of the 205, we could get a reliable estimate of brain health, or at least structural brain health, or at least total grey matter. So we measured total grey matter adjusted for head size, and that was our measure of brain health. And then we looked at whether the relationship between brain health and cognition was moderated by your midlife non-specific activities. Now, again, we did this at continuous interaction, but I'm going to show you the results split into 2 groups, high and low, a median split of those that did more than the median in terms of midlife activities, and those did less than the median. And here's the results. So on the y-axis is your current cognitive ability in old age, as measured by the Cattell test. On the x-axis is your current brain health, at least total grey matter, adjusted for your head size. And these are the 2 groups. The red group had low levels of midlife activity, and the blue group had high levels of midlife activity. And what you can see is there's a significant linear slope for those with low levels of midlife activity, and a non-significant slope for those with high midlife activities. In other words, the group with high levels of midlife activities, their cognitive health was less dependent on their brain health than those with low levels of midlife activities. Or say the other way around, those that didn't do so much outside the workplace in their midlife, their current cognitive ability was more dependent on their current brain health or grey matter health. So I think this then therefore makes midlife activities qualify as a type of cognitive reserve. So we identified a type of cognitive reserve, midlife non-specific activity, which predicts cognition many years later, 2 or 3 decades later in old age, over and above your education in youth and your current activities in old age. And it reduces your dependency of your cognition on your brain structure health. And it's potentially more easily modifiable by simple interventions than perhaps changing education. Now, of course, you may be wondering, what are these non-specific midlife activities? Unfortunately, the LEQ doesn't really give you sufficient precision to separate out physical, intellectual, and social components to this. There's only about 13 questions, and it's not possible to really get good measures of these independently. In other studies we've done in CamCAN, which I'm not talking about today, I would estimate that physical and social activities are far more important than intellectual activities. So seeing family and friends, doing sport is more important, I would guess, from other data than doing crosswords or playing an instrument. Anyway, there are caveats, of course, to this study. There's the main problem of what's called reverse causation. So we're assuming here that your lifestyle in middle age causes your cognition in old age, but it's possible that your cognition in old age affected your lifestyle, or at least your ability to remember your lifestyle. Or another possibility is that maybe your lifestyle was always influenced by your stable cognitive ability. So maybe certain people who have high cognitive ability choose certain lifestyles rather than the other way around. We couldn't control for that in this study, but I will mention that another study from the Lothian birth cohort by Gow et al., they have an independent measure of cognitive ability when these people were kids, 11, 12 years old. So they could actually covary out the cognitive ability at youth, and they still found a relationship between midlife activities and cognitive health in old age, suggesting it's not a stable relationship, that maybe that there is at least some part of the relationship comes from lifestyle to cognition. And arguably here in our data, although we didn't have a cognitive test performed in kids, I would expect such tests to correlate very highly with education, and we have got a measure of education. Another possibility is that people who are cognitively more able in old age are better able to recall or remember what they did earlier in their life. And of course, we can't rule that out either, although I would make a note that autobiographical memory, as in memory for what you did when you were young, is less severely affected by aging than, for example, encoding new information. We know as you get older, your memory to remember new things certainly gets worse, but actually autobiographical memory doesn't get so bad, at least until you get into dementia. And so I think that's probably less likely. But the only way to really get past these caveats is to have prospective studies where you objectively measure lifestyle, brain, and cognition in middle age and keep measuring it into old age. And then you can look at longitudinal analysis, lat analysis to see what is really driving what. And that's why I think cohorts like CamCAN are really vital to look at longitudinal study, but it's actually very hard to get funding for cohorts to make them longitudinal. It's much more exciting for funders to give money to a new cohort of interesting people rather than keep funding the same cohort for many years. But I think these are vital for science, to have prospective studies, longitudinal studies across decades, if possible, to measure brain, lifestyle, cognition. And the final thing you might be wondering, well, this cognitive ability that doesn't seem to depend so much on grey matter in those with high levels of midlife activities. It must depend on something in the brain. I'm not saying that it comes from some non-materialistic substance. So of course it may come from other aspects. So it may come from brain function rather than brain structure. And based on work done in Dallas by Chan and colleagues, I would suspect that functional segregation is a key variable that may allow some people to maintain cognitive ability despite grey matter atrophy. So as you probably know, functional segregation is how well different networks can remain separated from each other. And Chan and colleagues have found this relates to socioeconomic status. And maybe this is what mediates the relationship between cognition and the brain. And that's something we're now looking at in CamCAN, is functional segregation affected by midlife activities, and does this moderate or even mediate the effect on cognition? And hopefully have an answer for you soon. So with that, I'll shut up. Thank you very much. And this is a summary of what I've just said. I've got no idea how much time that took, but
we have a few minutes for questions, and then we'll switch to our backstage thing. But Rick, that was fantastic. It was a beautifully clear talk with absolutely wonderful results and some very provocative results as well. On behalf of the audience, I am giving you virtual applause. If I knew how to use the little symbols at the top, I'd give you real applause. But,
thank you.
Anyway, we're open for questions, and there's a question here from Dargan. which I'll just summarize. So the issue is that in CamCAN, resting state scans are done eyes closed. He says the MRI ones, at least, not clear about NEG.
Yeah,
whereas obviously, you know, when watching a movie, eyes are open. So the essential question is there a possibility that the comparison between the resting state scans and the movie scans are confounded by sleep, or the possibility that some participants slept and therefore disrupted their connectivity?
Yeah, that's a very good question. It's true that both the fMRI and the MEG were eyes closed. And I expect you'd get a different pattern of connectivity, or slightly different pattern of activity, if eyes were open and fixated, which lots of other people do use for resting state. It's possible that the differences we found between eyes closed resting state and movie watching was due to the visual stimulation. But there was also a third task called the sensory motor task, where people had their eyes open and they're responding to visual events and auditory tones, in fact, and pressing their fingers. And we also found in the paper differences in the effect of age according to that sensory motor task versus rest or versus the movie. So it's possible, I'm sure that visual input does play a role, but I think even if you had visual input in two tasks, you'd still find some different effects of age on those two different tasks.
Thank you. So another question, as you know, I personally greatly admire the work that you've done looking at vascular factors and their role in brain functional activity as a function of age. You described a very elegant DCM approach to this. I was surprised that you didn't mention the work you've done more recently on resting state variability as a way of trying to control for vascular differences as a function of age. I'm thinking particularly of the very compelling paper you had in Psychophysiology recently. I wonder whether you'd like to comment on that.
Yeah, you're absolutely right. So there's another line of work that Carmen Setvanov has led, asking whether you can correct task-based measures of activation. Whether you can adjust for vascular effects by using data from an independent resting state scan called resting state fluctuation amplitudes, or RSFA. So RSFA is simply the standard deviation over time of bandpass filtered fMRI data at rest. And the idea is that voxels that have higher variability have, if you like, more responsive vascular system. And Carmen's done some work showing that RSFA does change with age, and that if you adjust your task-based activations in old versus young according to independent data of RSFA at each voxel, then the age effects are dramatically reduced, although some remain. So he's got a couple of papers that show that in our sensory motor task, the visual effect of age that you see with a normal fMRI analysis disappears when you adjust for RSFA. Whereas the effect of age on ipsilateral motor cortex when you're doing a finger press, that effect remains even when you adjust for RSFA, suggesting it is a true neural effect. So RSFA allows you to adjust task-based activity. And to start with, we assumed in CamCAN that functional connectivity, as measured by the Pearson correlation coefficient, would be unaffected by sort of overall scaling effects in the vascular response, because it's a correlation coefficient, so it shouldn't matter too much if you scale either the data in either ROI that you're correlating. So if it was just a linear effect of scaling, RSFA won't help you with functional connectivity. In other words, if you adjust each voxel by its RSFA, it won't affect the Pearson correlation coefficient between voxels. But what we found in that Gillick's paper mentioned is that if we measure vascular health, the measures of functional connectivity do depend on that. So vascular health must be having some other effect, in fact, some nonlinear effect, which is what DCM is capturing. So yes, RSFA, good for adjusting task data. I don't think it's sufficient for connectivity data.
That's great, thank you very much. I think we should switch from the Teams the town hall meeting format to our round robin format. If people who are interested in continuing this discussion for a little while and allowing Rick to actually see them, would like to click on the link that's in the chat box, and Rick, if you could do that as well.
I can't.
Hopefully we can reconvene.
Mick, I can't get rid of my screen. I don't know how I stop sharing.
Lauren will help you with that.
Okay, thank you.